Okay, next we'll start product lifecycle management. Before going to this, we'll brief about guidelines, ICH guidelines, International Council on Harmonization guidelines. Uh, for ICH Q8, it's talking about pharmaceutical development, and ICH Q9, quality risk management, and Q10, pharmaceutical quality systems. By adopting these guidelines, we have to prepare our in-house standard operating procedures and standard test procedures. In this product lifecycle, we have to talk about QTPP, quality target product profile, and different quality material attributes and critical process parameters and critical quality attributes, and assess risk assessment, formulation process characterization, Q&Q composition by quality by design, design space development, control strategy establishment, product quality management, and regulatory commitment, drug product chain management. So, what are these? First, quality target product profile. It's a prospective summary of quality characteristics of the drug product that ideally will be achieved to ensure the desired quality by considering safety and efficacy of the product. That means you have chosen a product. That product should be I'm talking about generic formulation, ANDA. Your quality attributes must be same as innovator in terms of efficacy and safety. Okay, so in this formulation, what are the first you have to identify what are the material attributes, any critical material attributes affecting the CQS. What is my composition? Any critical materials are there in my composition, or you have to check it first. Then identification of critical process parameters. For example, it's a top spray granulation technique. In this top spray granulation technique, you are using some non-aqueous granulating fluid, for example. So this non-aqueous granulating fluid is having any criticality while spraying on the product. So these and all you have to identify in this stage. How these CMAs and CPPs are responsible for affecting the quality attribute, you have to study. Okay, next, risk assessment, quality risk assessment. During initial stage of formulation, you have to perform initial risk assessment technique. My unit operations are feasible to perform in large scale, pilot scale, pivotal scale. In this way, you have to evaluate each and every stage study. Then you have to go for process characterizations. So my technology is suitable for any other phase study characterization wise, it is okay. So it's a process friendly, process feasible, shop floor friendly. In this way, you have to evaluate, thorough evaluation is required. Then you have to finalize a formulation and process characterization. So by considering all these, before going for this Q&Q composition, this process characterization, if this process characterization raw material feasibility with the process are friendly or not, you have checked. Before this Q&Q composition, another one more thing you have to evaluate, that any safety related issues are there with the raw material, means not only API, any other raw materials. You will be checking the MSDS, Material Safety Data Sheet. By evaluating the MSDS, you will be understanding that my all raw materials are safe. So minimal precautionary procedures are required to develop the product. to manufacture the product. Like that, you will give some confidential statement. Then we can go further. Q&Q composition completed. Then quality by design. In this design of experiments, DOE studies will be done, which are nothing but a process optimization trials, formulation optimization trials. All these trials shall be finalized by QBD technique, quality by design technique, with DOE, design of experiments, which is having some software. That software shall give you the guidance for how many trials you have to perform, what are the minimum maximum specification ranges of that particular trials. Like that, it will give some brief idea. Based on these trials, you will develop a design space, which is called design space development. So initially you have a knowledge base you have. Out of that knowledge, so you are now n number of options you have. From that, you which option is adaptable, suitable to this particular manufacturing process, you have to identify in this design space development. So specification limits you are giving in this design space development. After design space development, control strategy establishment. So you have designed the process. What are the control strategies? Example, while preparation of a liquid oral suspension, addition of preservatives. Addition of preservatives are very important in this. I'm adding some X preservative in 2 minutes. I'm stirring this preservative in the main suspension for 20 minutes. Stirring is there. 3 minutes addition and 20 minutes stirring. 320. 320. So you have to establish control strategy wherever you go. So you are developing this process in lab. You are going for pilot plant. There also you have to add in 3 minutes. You have to stir for 20 minutes. Again, you are going for next large scale production. There you have to add in 3 minutes. You have to stir for 20 minutes stirring. In this way, everywhere these 3 minutes, 20 minutes should be constant in all phases. That you have to ensure. Next, product quality management. In the process of these development procedures, slowly, gradually, you are going to the large scale GMP area. From there, you have to follow all quality systems. It doesn't mean that in R&D you should not follow quality systems. We have to follow, but it's a non-GMP area. So trial and error methods, lab notebooks, writings, raw data entry into the notebook. So these and all required. So this is the developmental processes which is non-auditable. Auditor will not check all these documents like LNBs. Simply they will check a product development report, PDR. While coming to the PDR, you have to ensure all the quality management techniques, adopting these techniques into the plant. That thing you have to ensure. Next, regulatory commitment. For example, you are preparing a formulation for USA and Europe. So you have to follow both US FDA regulatory guidelines and European medicine regulatory guidelines. For these both regulatory bodies, you are filing a product at a time. So how you will do? You are preparing 2 batches. For example, my batch size is 5 lakhs batch size. I'm filing at a time for both markets. What you will do? Your manufacturing procedure is same. For Europe, separate manufacturing procedure. For US, separate manufacturing procedure is not there. Your manufacturing procedure is same. So you have to manufacture at a time. All the unit operations, all the product criticalities, everything you have freezed, you are doing in single shot. USA, as per USA, your COA is released. What about Europe? For Europe, all tests are may not be same. USA contains some microbiological study. Europe, it is not required. Some stringent impurity tests are there in Europe. So you have to perform that tests. Physically, all operations you have done in a single shot. For one batch, you are extending for European market. As per European requirement, you have to perform the analysis. So you can release Europe market COA also. So in your hand, USA COA is there, European COA is there. Your product is ready for filing of both markets. You have a data of US market requirements, European market requirement data you have. 6 months accelerated stability data is with you. Then you have to go for filing. So US body, US regulatory affairs of US team shall be file this product to US market. Europe regulatory affairs team shall file this market to European regulatory market. So separate filings are occurring. So your product cleared in Europe market. Now they have given some comments in European language, European nomenclature, European point of view, which shall not be applicable to US. This is what the commitment. So you have given some commitment to Europe. You have to follow that procedure for Europe only, not for US, which is a market specific formulation. You are formulating market specific formulation. For example, rest of the world ROW market, these and all are not required for ROW. In that way, you have to prepare for ROW market specification. So what I'm trying to say is, in this regulatory commitment, market specific requirements are very, very important. You have to fulfill that market requirement specifications. Then only you will get approval for manufacturing and production. In this way, the entire drug product chain shall be managed from development to marketing. Patient administration means so many intermediate steps are there. All these steps are meeting acceptance criteria of the innovator characterization. So this product life cycle. Once again, I'll tell this flow chart. QTPP, quality target product profile defining, identification of CMA, CPP, and CQA, and risk assessment Q9. Either a quality target product profile, you have to follow Q8 pharmaceutical development all guidelines. Risk assessment Q9 guideline, ICH Q9 guideline. Formulation process characterization and Q&Q composition by quality by design and design of experiments. Design space development, control strategy establishment, product quality management, quality commitment, regulatory commitment, and drug product chain management. In this way it will be. So Q8, Q9, Q10 guidelines are must while in this complete process.